view.
Hello and welcome to today's Aquarium at Home live stream. We are looking out the window of one of our aquatic biologists, Dr. Josh Ennen, and this is the view from his office at our Tennessee Aquarium Conservation Institute freshwater science facility on the banks of the Tennessee River. So this is what Josh gets to see pretty much every day when he turns around, but a lot of his time is actually spent
Looking out the window?
Probably looking at this computer screen,
Yes.
Doing a lot of science. So, Josh, first of all, thank you for joining us for today's live stream. Thank you to all of you who are watching. If you have any questions for Josh about what it takes to be a scientist, what science involves, or just science in general, or heck, he probably can answer questions about just about anything. Type them down in the comments. I'll pass them on to him. But in the meantime, Josh, tell us a little bit about yourself, what you do here at the aquarium, and kind of how you got started in the field of science.
Well, I'm the aquatic conservation biologist for the Tennessee Aquarium, and my area of study really is reptiles and amphibians, in particularly for the longest part of my career it's been turtles and in general. But here, since I've joined the aquarium since 2014, I think that was my first year, I really started putting a good group together, collaborators, John Davenport at Appalachian State, Kristen Sakala from Sewanee, and we all work with salamanders, looking at how climate change could affect salamander communities here in the southern Appalachians. So those are the two big study areas that I have, turtles and salamanders here at the Tennessee Aquarium. And yeah, day to day, if we're not doing field work, which usually entails either collecting salamanders for our stream experiments or collecting turtles in the wild, which usually runs in the summertime for turtles, but we still do quite a bit in the fall and into the winter months collecting salamanders and collecting data. But a lot of the time, when you think about scientists, a lot of your time is spent in your office crunching numbers, writing. Really, the end product of science is writing a scientific paper that gets published, and you can see that I got a nice little setup thanks to the Tennessee Aquarium. I got a nice little stand-up desk that I can operate with. Mm-hmm. But yeah, most of the time you're in your office, and it gets that way. The further along you get in your career as a scientist, as a field biologist, really, the less you're in the field and the more you're in your office. But there's still great days I get to go out, and one of my projects I have going on right now is really, if you come look, right down the Tennessee River up on the Cumberland Plateau, I have a couple of ponds, about 12 of them actually, at Prentice Cooper State Park. They're salamander ponds where I study a lot of stuff about embysthematic salamanders. And my collaborators on that project are Tom Anderson and John Davenport, Appalachian State folks. So I still get out, and that's an awesome study site because it's less than an hour drive. So I can get up there anytime I want during the day and check traps and things like that. But yeah, I mean, this is not the glamorous part of my job, but it's the part that you have to do.
So, Josh, which side of science would you say you enjoy more? Do you like to be out in the field more, or do you enjoy the time that you get to spend, on that most important of your tools, writing the papers that hopefully will end up in peer-reviewed academic journals?
It's a combination of both, I think. I think you have to have a love for both to do it and do it well. We all enjoy, especially days like this. It's really hard to be inside and you feel like you are in a cage looking out because it's such a beautiful day and it's nice and cool out. It could be a great day to be on a boat catching turtles. It would be a great day to be in a swamp looking for about anything. But you know that some days you need to stay in and crunch numbers because really you're out collecting data. You don't know the answer until you bring it into your office and you run your models and you look at what the answer is. And that's really what being a scientist is all about is you have a question, and you have to formulate hypotheses, and you have to test those hypotheses, even here with mesocosms or living systems inside the laboratory or out in nature trying to answer these questions. And one thing I really like about being a scientist is that you get done with one project, and it leads to 3 more. So it's like your job is never done, and there's never an insight, and it's one of those things when I talk to my family about what I do, why I do it, and what I do, it's one of those things I think they know me is that there's just no end to it. There's always more questions to be answered, and that's really, I think that's what drives me the most in science.
Every answer leads to more questions.
Oh, yeah, I love it.
So we do have some comments coming in. We got some shout outs. Looks like my mom is watching from Cumming, Georgia. So thank you for that. And
I played basketball with a guy from Cummings, Georgia.
Looks like your daughter must also be watching. Brittany is watching. She says, "We love you, Daddy."
Yeah.
All right.
And it is my wife's birthday, so I need to say happy birthday, Brittany. We love you.
All right. Speaking of questions, so as you're watching, please, looks like we got a donation. Thank you very much for that. If you feel if you have the ability to donate, and if you're in a position to, obviously we could definitely use the money at a time when we're not able to welcome visitors into the aquarium. Every little bit counts. So thank you very much for that. But speaking of questions, as you have questions, write them down in the comments, and I'll pass them on to Josh, and Josh will hopefully get an answer for you. But Josh, talk a little bit about where we are. So this is a relatively new facility at the aquarium. It's not anywhere that you've probably been. If you've been to the aquarium, this is our Conservation Institute. But talk a little bit about, what is in this building, and maybe we can take a tour while you talk.
Yeah.
And, why did we build this building?
Well, the aquarium built this building to I always tell people, put your money where your mouth is, and that's what the aquarium did. One of their mission statements is to protect freshwater biodiversity and habitats here in the Southeast. So they built a building, and that's why they hired me, Dr. Bernie Kahida, and Dr. Anna George, Sarah Sweat, and Shauna Fix. She's just changed her name. She got married, so I almost got caught up there. She was going to kill me for that one. But we have this group of biologists, and that's what we do. We try to protect freshwater animals and protect their habitats. So Shauna, she sits right here. She's socially distanced at home. She works with Laurel Dace, a critically endangered little minnow up on Walden's Ridge. So she does a lot of great stuff. And then if you turn around, if we come to our GIS suite, as I like to call it. I told Sarah she doesn't have it all, but she has a suite. And that's Geographical Information System. And this is where you put dots on maps, do spatial associations, look at kind of habitat parameters. And this is really pivotal to what we do here, is look at where and why endangered animals are located. And that's one of the fundamental things in ecology, is trying to figure out where and why animals occur where they are. And Sarah's critical to that. And you can see this map is from a project Bernie and Anna and Sarah did on mapping out biodiversity. So this is kind of what GIS is in the end result. But it really entails a heck of a lot more than just putting maps together. Sarah's phenomenal.
Yeah, the great thing about these kinds of maps is that a lot of science really is crunching numbers and looking at data. And what Sarah does that's so interesting is she takes that and she makes it digestible. She gives it context, makes it something that you can look at and really see. In this case, we're looking at a heat map. So this is showing the number of, or the density of species found in a particular watershed. Yep. So the warmer the color, so the closer it gets to red, the more species are found.
More mussels and more crayfish you find in those watersheds. So you can see the mobile drainage is really important for biodiversity, and then the whole Tennessee River is very important, and then it's part of the Cumberland as well. So those are the three really important rivers for biodiversity in those three taxa.
So in essence, if you live in Chattanooga, if you live in this area, you're living pretty much at the very center of one of the richest aquatic ecosystems in the world, really. Yeah, I mean, this is one of a really great place to do science, I bet.
It is a biodiversity hotspot. For those three groups that we talked about, it really is a biodiversity hotspot, in particular mussels and crayfish. There's no other place on the planet that rivals the numbers that we find in the Southeast, and the same thing can be said for salamanders in the Southern Appalachians. We have more salamander species in East Tennessee and Western North Carolina than most states combined. Turtle-wise, the state of Tennessee has over 16 species of freshwater turtles. And I know that doesn't sound like a lot, but there's not that many turtle species globally. There's been a couple described here recently, so my math may be a little off, but it's around 357 species globally. And the mobile drainage, so right down here where this hotspot is, they have more species of turtles in the mobile drainage than almost any other river system in the world. So we do live in a place where you can walk into your backyard and just know that you're in a biodiversity hotspot, and it's very special. And just know that you live in the watershed of the Tennessee River, and that's on this map that we just showed you. It's blood red because we have so much wildlife here aquatically and terrestrially too, if you want to bring that up. But so if you want to keep tabs and keep sure the water are healthy, especially for turtles, you don't want to take away any of the riparian zone vegetation. And all these things you can do for turtles will benefit fish, mussels, and crayfish as well. Take care of the shoreline. Make sure you leave snags down in the water for turtles to bask. Make sure, you keep some parts of your yard wild for some of those terrestrial box turtles, which is the state reptile for Tennessee. And then we have a couple of those on exhibit here as well. So yeah, it's just a great place to work and live, to tell the truth. And I grew up in the South. You can probably tell through my accent. So I have an affinity for the South just because it's a home. I've lived in South Carolina, I lived in Louisiana, I've lived in Mississippi and Tennessee. So I've been all over the South, and all of them are unique, every area that I've lived. But I tell you what, Chattanooga, Tennessee is one of my most favorite places I've ever lived, to tell you the truth, in the South. It's beautiful.
So Courtney Keys asked the question, and this may be a hard one to answer, but how many turtles do you save a year?
Save a year? I'll go with all of them. I like saving them off the road. I'll pull or move turtles off the road when I see them. I don't know what kind of a number. I don't see as many. A lot of times when I see box turtles are usually when I'm going up to Prince Cooper and it's on a gravel road, and I may see maybe 4 to 6 of those a year that I'll move off the road. And if you move a turtle off the road, make sure whichever direction it's heading, move it over there and do it in a safe manner where you don't get run over by the car on oncoming traffic. So yeah, saving turtles in that sense, there's been a couple times where I've removed fishing line from turtles, stuff like that, but that's over the years. It's not really like I could say I do that every year. But I think in a small part, I think that everything I do at the Tennessee Aquarium is saving turtles. We do a lot of global conservation work. Me and Sarah Sweat looking at where and why turtles are where they are. Can we prioritize regions so we can put more conservation dollars there for the global community to come on board and protect turtles? So really, I think hopefully some of my research will save a lot of turtles in the long run. So me and Sarah have done a lot of good stuff with spatial, identifying global hotspots for turtles. We're using turtle data.
All right. Well, since we are in the Conservation Institute, we have seen thus far a couple of offices that maybe don't look particularly sciencey, but this building does have some pretty state-of-the-art facilities that probably will scratch that science itch for you. If we can take a look real quick at some of our labs, and you can tell us a little bit about the kinds of work that goes on in them.
Yeah, follow me. So this first lab, everybody put your food up, okay? So there's no food or drink. So this one here, this is our genetics lab and microplastic lab. So what does that mean? Here you can take tissue samples, could be a small fin clip, a small hair sample, it could be a small, anything that's vascularized you could take, get DNA from. And here we kind of look at conservation genetics. Okay? Are there unique populations within an endangered animal? So should we treat this population on the north end of the range? We should conserve that a little bit different than the south. Because if you have all this genetic diversity, you want to conserve that, because the more genetic diversity you have in a population, the more likely it can adapt to future climate events. So we're really concerned about genetic diversity. We do that same thing with our lake sturgeon program, our brook trout program, our anything that we propagate downstairs. We want to look at genetic diversity, make sure that we're releasing propagated animals, hatchery animals that have a representative of the total genomic makeup of the species in general. So this is what we can do here. We have a lot of PCR machines and all the stuff.
What is PCR machine? For those of you who are watching who maybe don't know.
That's I always explain to people. Anybody can do PCR. I was never very good at it because my PhD advisor always said the PCR gods were not happy with me. But it's like you have a recipe, you follow it. It's called a thermocycler. You put it in there and you have some enzymes that actually replicate and reproduce the DNA over and over and over. So then you can amplify it and look at it, sequence it, or you can do microsats depending on what kind of loci you're looking at. So that's what goes on here. And then to your left is our brand new microplastic microscope. So this machine, if you take a water sample from the Tennessee River or anywhere else, it will tell you kind of how much of the particles there's, kind of abundance or density, and it will also tell you what kind of plastics are in the water. So And one thing we have going on globally is we're drowning in plastics. We make more and more plastics every year, and what we're finding, and most of the research has been done in the marine environment, is that these plastics break down over time, and we're starting to get these tiny, microscopic pieces of plastic that at the bottom of the food chain, these animals will uptake that. We'll eat it, we'll mistake it for food. So now some of those can move up the food chain, and you can get online and you can see all sorts of things that consume microplastics or plastics in general. So the Tennessee Aquarium is taking a stance on this, where we have trying to go to zero plastics. You can see that our such a stance. We don't have straws anymore. If we do, they're paper or metal straws. So we're really getting away from plastics, and we're really putting our money where our mouth is, and trying to study how all these microplastics, how they're going to affect animals in the wild. So this is our kind of our micro, if you will, lab, genetics and microplastics. And then our next lab we'll look at, we call it our morphology or ecology lab.
Now, before we go in that, let's answer some of these questions that have come in, because they have been stacking up a little bit. Bernie, I'm going to guess it's Kahajda, would like to know, what is your favorite turtle?
Favorite turtle? If I ever get to name one again, I'm going to name it after Bernie. And then that would be my favorite turtle, Bernie.
Bernie Kahaya is our manager of science programs.
And my boss.
But really, Josh, do you have a favorite turtle?
Favorite turtle? I don't know. My wife gets mad at me too. She always asks me, "What's your favorite meal?" And I'm always like, "I don't know. I haven't thought about it. I like all food." But so it's kind of like I like all turtles. But I guess I'll go with the group, Bernie. I'll go with map turtles. I like graptomes. They're a southeastern species, and there's a lot of, this is for you, Bernie, a lot of narrow endemics. Those are species only found in a very small, restricted range. So a lot of the species that I worked with for my PhD were narrow endemics that were only found in one river system. And I actually described a new species of turtle back in 2010 in the Pearl River. So it's only found in the Pearl River in Louisiana and Mississippi. So I'll go with all map turtles. I kind of like those.
So before we go to the next question, again, this is a lot of really good science vocabulary. If you're watching at home, this happens to be your science hour if you're doing the homeschooling that most of us are probably trying to do right now. Can you tell us what describing a species means?
Well, a lot of work. So what I did, there was one species of graptomes in this larger group of graptomes called the broadhead. They have a broader head, and that's because they eat mussels and clams. So there was one species that was found in two river drainage, so I thought, well, that doesn't make sense. If all other ones are restricted to one, this one should be restricted to one as well. So I looked at molecular data, so I'd use a lab just like the one we just talked about, looked for certain molecular markers and see how different the two drainages were. So we did that, and we found out that they were different. To the degree where it could be a new species. And I'm kind of old school in the sense where I like to do molecular work as when you're doing these kind of things with describing species, but I also want to go back in and look at morphological features. So if you had these two animals in hand, what are the morphological characters that would distinguish them? So if you write
what physical characters?
Physical character, yes, like shell color, shell shape, size, head pattern, colors, markings. So you put both those together in some statistical analyses, and if you feel that it warrants a species description, you try to submit it to a journal, and then a group of your peers will tell you if it is or not.
And that's how we get new species.
Yes, that's for a lot of times you do. And we're in an era now, because of molecular data like the lab I just showed you can generate, that we have a boom in new species. So I don't want everybody to think that it's all doom and gloom when you hear of these extinction crises, which they are the case, but every year there's large vertebrates that are described. I think since I've been quarantined, there's a new species from the Amazon, from the Mata, one of my favorite species, an ambush predator from the Amazon. They split that into two, so that's pretty cool.
All right. So now that we're in this other space, obviously we're seeing a little bit more of the science-y kinds of tools and of the trade that you would imagine when you think about somebody being a scientist. So what happens in this lab?
A lot of things going on. So this is Shauna Mitchell's area. She marked it off nicely with orange tape. So she's looking at otoliths, which is a bone in fish in their head that you can actually age like it's a tree ring. So she's looking at aging all her fish species that she's catching with the Tennessee days. You can take measurements here. We have microscopes that you can count scale rows to identify fish, but you can also look at tooth rows. You can look at all sorts of things in tadpoles, but you can also look at diet content. We had one intern for our George Benz Fellow, it might have been 3 years ago, that looked at gut content of a minnow on Walden's Ridge. So this is where that will happen. A lot of stuff that you collect in the wild and we'll bring in here to look at answer those kind of questions. And so we have stuff just scattered here throughout. We have our water parameter machines. This is where we do water quality for everything downstairs. We put that up here. So this is where all the propagation staff comes up and make sure all our water quality for our fish downstairs is primo. So this is where this happens. And so, I mean, we don't have much going on in here right now just because of the quarantine situation. But I spent a lot of my time here. A lot of the jars that you just saw with fish in them. So in science, there's a lot of collection that goes on, not really like it used to be, but when you go out and sample, there's fish collections dating back, and Bernie could answer this in the comments, that go back way back. I mean, beyond the 1800s, you'd probably find fish collections, especially in Europe. But what we have is, when people go out and collect fish, it's important to collect these fish and have these records in museums so scientists like myself and Bernie can go study how fish morphology have changed, document distributions, describe new species. I go back and work in museums for turtles all the time. I work a lot with Jim Godwin and all the folks down at the Auburn Museum. Auburn University has a museum there, and they have a huge turtle collection. So I've written, I don't know, maybe 3 or 4 scientific papers on turtle morphology based on turtles that were collected and harvested from the 30s on into the 60s. So it's valuable data that are in these institutions all around the globe, and they're vital.
All right. Nikki Cox would like to know, what can you do at home to encourage and support turtles in your backyard?
Leave sections of your yard wild. And if you want, you can get online and do some research and plant certain plants that native terrestrial turtles like. So, for example, one of the best native terrestrial turtles we have is a box turtle. And they are really huge in seed dispersal. So they like eating fruit. So you can plant a lot of wild things, like wild strawberries they'll eat, wild blackberry bush would, if it's low enough to the ground, they'll consume those. So they're one of our really good terrestrial seed dispersers here in the native. May apples is another good native plant that they like to eat those as well. So anything like brush piles, anything like just let it go wild, and that's good habitat for them to overwinter in and seek shelter. And you got to know, if you have a box turtle on your property, just know that their home ranges are not that really large, relatively speaking. So they may be confined to your backyard or your neighbor's yard or 2 or 3 neighbors. So whatever you can do to help them in your yard will increase the benefit that you may see baby turtles in the future in your yard, which is great. We had in our backyard, I live up on in Signal Mountain on the backside. We had one turtle that my son, when he was little, named it Morty, and it was a box turtle. And I swear Noah could flip a rock, get a worm, and if Morty was in the yard, Morty would come up and eat a worm out of Noah's hand. So, and he always wanted to bring it as a pet, and I always told him, no, let's leave wild things wild. And so I let him put a little dot of Mama's nail polish on it so he could identify it for later. And we caught Morty for years and years and years. Yeah.
So KB Jones says, my 7-year-old asks, what kind of turtle does Tennessee have the most of? So what's the most abundant species in Tennessee?
Oh, anywhere in the southeast, it's going to be the pond slider, Trachomys scripta. They are everywhere. So that would be your most abundant turtle species. I did a project, a 3-year survey of turtles in West Tennessee, partnering with Rob Colvin from the Tennessee TWRA, Tennessee Wildlife Resource Agency. And yeah, the most abundant turtle species out there is the red ear slider.
Okay. Marilyn Payne, who is one of our coworkers, would like to know, what's the difference in salamanders near streams or creeks and those in wooded areas or backyards? Are they related?
Yeah, well, they're all probably part of Plethodonid salamander, the family, the lungless salamander. The ones that you'll probably find more commonly in your backyard would be Plethodonids. And the genus Plethodonid would be kind of your redback salamander or your slimy salamanders. And then the ones more commonly seen in the stream would be your Desmognathus, your dusky salamanders. So depending on what elevation you are, so if you're down in the valley, the most common Desmognathus that you would catch in the stream would probably be either the spotted dusky, which is Conanthi or Fuscus. And the further up you get, you'll get into the seal salamanders. You'll get into black bellies further up if you're in the southern Appalachians. And also some common ones would be your two-line salamanders. Those are really bright, yellow if you get higher up in the mountains, but down here there'll be more drab golden color, they're still pretty. And if you're lucky, you may see some red salamanders along the stream. They're one of my favorites, beautiful. So get online, you can get some images of those. And we go on our website too. We have, I think we have a place on the website we talk about Pseudotriton rubra, the red salamander. So those are the type of ones. So they're all related, but they're all genetically different depending on what family they are in.
Now, since Josh just made a very convenient segue here, I'll go ahead and tout the fact that on our website, tnaqua.org, we do have a lot of resources available for those of you who are sheltering at home, who are socially distancing yourself, because we have lots of different tools and resources that you can take advantage of to keep yourself or your kids occupied. We've got activity sheets, coloring pages. Actually, Josh has recently worked on an interactive story map with Sarah, who was the GIS analyst we were talking about earlier, to create a really cool interactive guide to some species of turtles all over the world.
It was Sarah's idea. Sarah has great ideas, and she has the wherewithal to do these things on the computer and post them online. And it's a story map where it's basically, if you're interested in turtles, there'll be a link. Casey can post it later, you can find it. But Sarah's idea was to kind of do like a virtual, not really a virtual tour of our new turtle exhibit, but kind of a interactive look at pictures, and then have videos embedded of all the turtles, not all the turtles, but most of the turtle species we have at the aquarium, or the most interesting turtle species, if you want to put it that way. And we put it together, and I wrote some content on basic facts and pulled some stuff that we already had off our website. You can go on there and learn about the alligator snaphead turtle, my project that we did in West Tennessee in collaboration with Tennessee Wildlife and SEMO, Southeast Missouri State, and John Davenport. You can go on there and you can see some stuff that Bill works with. Bill Hughes, our herpetologist that does a lot of propagating and hatching of rare and endangered species. It's four-eyed bills. We have a little section there. And we have a turtle on there, spotted turtle, which are beautiful turtles that are native to the Atlantic slope, which means they are the east side of the Smoky Mountains, if you will. So all on that coastal plain from Florida all the way up to Maine, they're spotted turtles. And Dave Collins, our director of husbandry, he does a lot of work with AZA SAFE program, keeping turtles safe from extinction, and that's one of the species that he's in charge of. So, I mean, if you think about the Tennessee Aquarium, this is how I think of the Tennessee Aquarium, and you should too, is that we really have turtles covered globally, and we have turtles covered locally. That's one of the reasons why we have one of the largest collections of turtles on display in the world, because Dave Collins and Bill Hughes have always had affinity for turtles. And like Bill always said, which is a great quote, he goes, it's always a year of turtle to him. And he's just glad that everybody came on board this year for turtles. So Tennessee Aquarium has always been synonymous with turtles in my mind.
So again, that website is tnaqua.org, and all of those resources are kind of gathered in one place under our subsite, which is our Aquarium at Home. And actually, Josh touched on a topic, so watersheds, while he's getting something out of his office, touched on a topic of watersheds and what watershed we live in. And that actually is tied to Weekday Wonders, which is a new series of curated content created by our education department that should get young people outside of the house, or I guess older people if you're into that, get them out of the house, into the backyard, away from their screens, thinking about the world around them in maybe a different way, and maybe getting some of that energy out that we're all probably getting pent up, getting stuck in the house. So go to tnaqua.org, go to our Aquarium at Home section, then look up Weekday Wonders, and that'll be a good place to get all of that information. So while we're walking around to the back of the building, I'll see if there are any more questions I can get answered for you. Courtney Keys asks, how much plastic is in the water? So this is going back to talking about microplastics.
Go online and look for tin swim. And there was a German scientist, Dr. Andreas Voss, that swam the whole length of the Tennessee River that has that data posted online. I don't know the densities and don't know the comparative the relative density to other drainages, but yeah, check that side out. And Andreas was here, and he collaborates with Dr. Anna George and Bernie Kahida on working with microplastics using that new microscope that we got. So this is really interesting. I park right here every day I come to work, and this is all the created wetland that we did when they built the building. And I know it doesn't look like a wetland, but it is. And every day, before I go into work, I'll stop and listen to see what new frog species. And we have a really good, there was a lizard. We have a really good population of coarse frogs that call, peepers that call, green tree frogs. And then in the other part back here, the really modified wetland that we created, we hit narrow-mouthed toads, which are really interesting. And there's a website that Tennessee, I think TWA still has, that you can go on and check out all the frog calls, and check out narrow-mouthed toad frog calls. One of the coolest ones.
So TWRA is the Tennessee Wildlife Resources Agency, if you want to look that up.
Great collaborators and friends of Tennessee Aquarium.
That's true. That we do work with them quite a bit. Lou Everman would like to know: do our reptiles share similar features with reptiles from tropical rainforests, since we live in a temperate rainforest?
I mean, we have some genera that go all the way to the tropics. So the pond slider is related to some tropical species. I don't know. That's a good question. Never really thought of that. Characteristics. I do know, I do a lot of stuff looking at reproductive characteristics of temperate and tropical turtles, and you get larger clutches and larger eggs the further north and south you get. So when you get into the tropics, they usually have smaller clutch sizes. That means how many number of eggs per reproductive event, so they have smaller numbers there and smaller eggs. So that's one characteristic that's kind of different. So there's a lot of cool latitudinal gradients that you can look up on clutch size, reproductive output, body size. Turtles towards the poles are usually bigger than your turtles that would be in the tropics. That's a general trend that occurs, and that occurs with a lot of different species. So this is another not temporary wetland, but wetland that we created. So all the water from the roof line comes down, and we pump it into this basin, and when it overflows into the concrete basin, it dumps into here, and this will fill up. So these are all native plant species that you would find in wetlands around the gorge area in Tennessee.
This whole building is LEED Gold certified, and it was purpose built with conservation pretty much from the roots to the rafters. I mean, conservation informed basically every step of the construction of this facility, which opened back in November of 2016. So we'll be 4 years old later this year.
So I did set some props up because I didn't know what else to talk to you all about. So I was going to show you how I would catch salamanders and turtles if I was doing my study. So come with me. We'll do salamanders first. So, I mean, you could use the basic net to net out salamanders or tadpoles. You can still see a lot of these are probably green or bullfrog tadpoles in here. But this is basic what we use when we look for bistemmatid salamanders. So in the ephemeral or temporary wetland ponds that you can find around here, and those woodland salamander species. So this is basically a minnow trap, and a lot of times you can use it to catch minnows as well. But it's just basically 2 funnels. You pop them together and put a clip on, and I always like to set them with enough air, breathing room, so that you don't drown anything. But this is a typical technique that a lot of people use to catch salamanders and small fish in swamps.
And there have been times, Josh mentioned Prentice Cooper State Park, where he does quite a bit of his salamander research, and we've been out with him before to cover that work, and these traps will come up, I mean, just absolutely packed with salamanders.
Yeah, these ponds, and like my buddy Davenport always says from Appalachian State, that's his springtime indicators when all the spotted salamanders start moving to ponds, and that's what we study a lot of: the spotted salamanders, marble salamanders, and mole salamanders. So this is a good way to technique it. So here's you see this? There's a little going. Yeah, we have a lot of frog species that utilize this for breathing. We get gray tree frogs, green tree frogs, bullfrogs, green frogs. So we get a lot of species that are utilizing these human-made wetlands.
And it's supposed to rain in Chattanooga tonight and through pretty much all day tomorrow. So, Josh, after a rain event like that, where a lot of water comes down, what will this area look like?
Oh, this whole area back here will be, a foot of water. When it comes, so basically this will fill up. Everything will come off the roof, fill this basin up. It'll come shooting out, and water will be spraying everywhere. It'll fill this up to the brim. It'll overflow, and then it'll fill this whole section up, and then it'll float kind of back up to the front of the building, and then down into the natural wetland that we modified to make it a little bit bigger. And then basically we did that because wetlands are a natural filtration system. So it filters all this water that you don't have to spend money on to filter at a filtration system. All these plants do that, taking off toxin and all that. And it just goes, and I mean, you saw how close we are to the Tennessee River, so all this water gets filtered, gets dumped into the Tennessee River.
Yeah, for those of you who are joining a little bit later, that ridge line is on the other side of the river. So we are very much on the banks of the river.
Yep, and this is your basic 3-ring hoop net. So this is the same kind of trap that I used in West Tennessee, looking to capture the alligator snapping turtles. Same trap that I caught mostly sliders in. Everybody jokes with me, I didn't catch many alligator snapping turtles, but I sure caught a lot of sliders, which most people do, trust me. Except if you get down south into Mississippi, I got a buddy named Luke Pearson that catches more, I think, alligator snapping turtles than anybody. But it's basic stuff. It's like the same idea as the minnow trap. You have a throat that you call, that the turtle climbs up and goes in. You have an area back here. These are actually baited. I always have a bait bag, and you can bait these. Out west, in West Tennessee, when we were doing the project, we did fresh fish because we were targeting alligator snapping turtles. But if I was going to trap in the Tennessee River up, I started want to trap in my ponds at Prentice Cooper. You could do catfish. Some people would do dog food, dry dog food. Some people can do chicken liver. A lot of people will do stink bait, what you would use for catfish. So you can use a lot of different things to catch turtles, but that's kind of the idea that we have used. And you can see that we have 2 metal poles opening the whole trap up to keep it open. If I were to take these off, the whole trap would collapse down. For easy storage. So there you go. I mean, that's all a trap, all to it.
So I guess if there's a takeaway from this, I mean, I think people probably have this conception of science as being lots of really high-tech equipment. I mean, obviously we have some of that. You've seen the labs that we've got upstairs in the building, and those have things like those spectrometers that we were talking about, and the microscopes and all of that equipment. But really, a lot of it is just fairly unsophisticated. I mean, that is not a very complicated bit of machinery, but it's the kind of tool that your work would be very hard to do without.
No, it's a lot of the field biology is just boots on the ground. That's the hard part.
Getting willing to get dirty
and enjoying it. Really, you can't shy away from mosquitoes and being uncomfortable for a couple hours. And after a while, you get kind of used to it, and you kind of miss it when you're not doing it.
There's some pictures of Josh I've taken of him pretty much up to about his neck in swamp water, and he's just got a big grin on his face. So
that's how I grew up. This is every day I get to do this is like when I was a kid. Because so when I grew up in Louisiana, essentially you can ask my mom if you ever bump into her. I'd be gone in the morning and go straight to the bayou, and I had buddies that we were either catching crawfish, we were catching snakes, we were doing anything we could do to be outside, and that's kind of being in the swampy waters is something that I've always done.
So going through a couple more of these questions that have come in, Ashley Sebastiani, I'm sure I mispronounced that, I apologize if I did, wants to know how many species of turtles are there, and you did touch on that earlier, but if you don't mind doing a quick recap.
Globally, it changes because there's been a few described. So we're right around 356, 57, 58, around there. That's just species. That's not including subspecies. So in the state of Tennessee, we're right at 16. Most of them are found in West Tennessee. But in the United States of America, it has the most turtle species in any other country. It's a huge area, obviously. But the Mobile drainage in Alabama, that drainage has more turtle species than anywhere else on the globe. It's comparable to the Ganges. So the southeastern United States is really a turtle hotspot.
Okay. Meredith Harris, who actually is another one of our Tennessee employees, so Tennessee Aquarium Conservation Student employees, she wants to know, are some endangered turtles being reintroduced, and what are some challenges facing a program like that?
There's always been reintroductions. A lot of translocations going on with turtles. So a lot of tortoises, terrestrial tortoises, especially gopher tortoises in Florida and throughout their range in the southeast and the coastal plain, they get moved a lot. Desert tortoises get moved a lot. There's a lot of head starting and propagation going on with gopher desert tortoises. TWRA, Tennessee Wildlife Resource Agency in the state of Tennessee, are thinking about reintroducing alligator snapping turtles. So a lot of that has to do with hatching them out, raising them up to a certain size, head starting them, because turtles at a small size, from eggs to about, box turtle size, are just food for everything. So most eggs that a female lays in her life will be consumed by another animal. So that's where the head starting program comes for a lot of turtles is, well, if we raise them up large enough, then they can't be consumed. And that's what a lot of people are doing for turtles throughout the globe, really. There's conservation efforts going on with one of our partners, the Turtle Survival Alliance, that they do this at a global scale at certain places where a lot of endangered turtles are located. So the challenges are what you would see everywhere. It's hard to work with long-lived animals. And a lot of research that's come out suggests that you need to release all age classes when you release turtles when you do that. So there's a lot of pros and a lot of cons when it comes to releasing turtles in the wild, but you have to do something, and a lot of people do it. And the one population that we found in West Tennessee was a reintroduced population from TWA 10 years ago. So it worked, and we caught, I think, around 17 of them at one site that were all introduced in 2005.
Mary Ann Williams would like to know, do you see Pigeon Mountain salamanders often?
No, I have never.
Never?
No.
A salamander that Dr. Josh Ennen has not seen?
Yeah, no, that's only found in one small location. No, there's a lot of salamanders. There's one that's fairly common in northeastern part of Tennessee, Yonah-Lussies, that I have never seen. I just never made it up there.
Amanda Howell says, love your informative, fun videos. So grateful you all do this. Well, thank you, Amanda, for saying that. We are doing this because we want you to feel less confined, get some good information, feel like maybe the walls that surround you are a little bit farther apart than they actually are. Keep you connected to our animals and some of the work that we're doing. So thank you for saying that. Thank you very much. Looks like we've got a few donations that have come in as well. So thank you to those of you who have donated. That button is there on the screen if you are in a position to make a donation. Obviously, the science continues, the work goes on. The animals are still being cared for, and along with that comes some inherent cost, and anything that you can do if you're in a position to help defray that cost is just very much appreciated. And if you can't donate, that's fine too. We are still here, we're still doing this, and we love to be able to give you guys an insight into what we do. So
if you don't donate, come visit us when we're open again.
Yes, come visit us, and in the meantime, what you can do is share this live stream with your friends. Make sure that as many people see it as possible, because maybe one of your friends is in a position to help us out.
You want to see some more stuff?
Yeah, let's go see what else we've got here.
So this doesn't look like much because we obviously with the quarantine situation, and it's getting a little too hot. So in this carport, this is all our we call them mesocosm. What that is just artificial streams. So when these are hooked up, they don't look this messy, but what it basically is, we'll have a flow through system. So we'll have within these 4-foot tanks, we'll have running water, and we'll have cover objects of what you would see in the wild. And in here in these systems, we've done a lot of phenomenal research with headwater stream vertebrates. What I mean by that are these small streams you see way up in the southern Appalachians, where you have very limited amount of fish that occur up there, and the most dominant vertebrate that are predators are your salamanders. So we look at these kind of unique communities in the southern Appalachians, and we try to see how variability and climate is going to influence competitive interactions. And a lot of the species that we use are blackbellies, seal salamanders. We use some of these smaller desmogs that are more terrestrial in here as well. We've used red salamanders, but we also pair them up with fish. And our idea is to look at if we raise the water and increase the flow and try to mimic kind of a huge rain event, how does that affect the interactions between animals? And so we do that more often, kind of like the frequency thing that you see now. We see more 500-year flood events, and that's kind of what we're trying to do, is see how are these extreme weather events going to influence our communities here in the southeastern United States in high elevations. We also look at temperature effects, and one of the cool results we found, this was with a student, an undergrad from the University of South Sewanee, one of Kristen Sakala's students. We actually published several papers with Sakala's undergrads, and what we found was that if you increase water temperature, it actually changes the strength of the competitive interaction. So what does that mean? Well, usually under cooler climates, the bigger salamander dominates the smaller salamanders. But if you increase the water temperature to a level that is not natural, the smaller salamander actually is able to cohabitate and actually grow and put on body mass. So we're doing a lot of really cool stuff in these small systems that don't look like much right now. But yeah, it's very important work, and we have a lot of good collaborators to go into this. And that's one thing that I think a lot of people don't realize. It's not just me sitting in an office writing papers or me in the field. Science is a group effort. I always love that the saying, I'm paraphrasing, that it's kind of stand on the shoulder of giants. What that really means is you just build on each other. You build on the data somebody else collects and you get more ideas. But it also means, in my mind, is that you have these collaborations, like with Sewanee, with Appalachian State, with TWRA. You can't do all this alone. And that's one thing that science is. It's a community, really, and it's all of us together working to answer questions and find out more questions to answer. So yeah, I just wanted to mention that. So science is really a collaborative thing.
So Josh, you've been here at the aquarium since 2014, you said. So this is 6 years you've been doing this. Mm-hmm. What made you, and I'm assuming you had experience as a biologist before that, but what made you initially want to become a scientist?
I was really young. I think I spent a lot of time outdoor as a kid, and I've always been curious and question driven. I think my mom could correct me on this, but I think even when we were still living in Illinois, that's where I was born. That's what trickery, right, with my accent. But she always said that I always said I wanted to be a scientist or a pilot and stuff like that. So I've always had a curiosity about nature. So I go to nature and try to learn something. And I guess that just kind of, as I grew up, it stuck with me. Because as soon as I went to college, I kind of think, I wanted to do biology. And like I always said, this is kind of the stuff that I did as a kid. Go outside, play in the bayou, catch crawfish, looking for snakes. So this is kind of extension of childhood, really.
So what advice would you offer to somebody who maybe is watching this, and if they're younger and they have a similar, interest in being outdoors and or have a curious nature, what advice would you give to them about what it takes to become a scientist? What should they be doing now?
I just keep your mind active, be outside, and when you find stuff, look it up. Like, one thing that I always wish I would have done more of is identify plants and stuff like that. But I was always real curious about put a bird feeder up, and I'm teaching my kids birds. Flip over rocks, and if you see something you don't know, take a picture of it and then go look up and see what it is. So it's just basically becoming a naturalist, being curious about your surrounding. Some of the greatest discoveries in science, I think, come from just mere observation of something in nature. I had a good friend that he wrote a whole really sweet paper about competitive interaction between plants and animals, and it was just basically he was aware of his surroundings when he was in nature collecting data, and he's like, ooh, this is a cool observation. Is it true? So that's the other thing, just be curious and be aware and observe nature. And I think that's where you can get a lot of your curiosity, and a lot of the scientists come from that kind of background. But like I always tell, everybody always asks me, are my kids going to be scientists? And I always tell people, I raise my children, I want them to be happy so that on Mondays when they go to work, they're not miserable. Like, I love Mondays because I get to do this. So I want my kids to do stuff that they're happy with, but I'm raising them so they're raised by a dad who's a scientist and a biologist. So if they go on to be whatever, really, I don't care what they become as long as they're happy, but they'll be informed and love nature, and a lot of their decisions in life and their personal choices will reflect that nature is a part of the whole community, the whole global experience. If we don't have nature, we don't have an economy. If we don't have nature, we really don't have much. You can't, it's really everything we get from is driven from this globe. So we have to take care of it.
Well, and that's an excellent message for Earth Day. I'm not sure if I actually mentioned the fact that today is Earth Day, but thank you for joining us for today's 50th anniversary of Earth Day live stream. Josh, thank you for telling us about science and why you do it, what it entails. Obviously there's great value in the work that's being done here at the Tennessee Aquarium Conservation Institute. To all of those of you who have joined us, thank you very much for sticking out with us and taking a tour of our Conservation Institute here on the banks of the Tennessee River. To those of you who have donated, we cannot express just enough how meaningful that is, how important your contribution is, and the good work that it will be put toward. Yep. We certainly appreciate it. And those of you who've watched, hopefully you've learned something. If this is your science hour, hopefully you walked away with some new words to look up. Josh is definitely a font of some pretty big science vocabulary.
Well, my wife wouldn't agree. My vocabulary is very limited, she thinks. But I do want to say hello to everybody I'm related to who's watching. And my cousin Andy, I wore a collar this time for you. So that goes to you, buddy.
All right. Well, on that happy note, we will end today's live stream. Thank you so much for joining us for this issue or episode of Aquarium at Home. Tomorrow we will be back at about 1 p.m. Check our Facebook page at about 11 a.m. to find out what we will be talking about and where we will be. And again, thank you very much for watching. We will see you next time.